to be doing our first build a lecture on our microbiology class. I chose to do chapter 5, which are the eukaryotic cells and microorganisms. So it goes into detail about definitions, the processes, the stages, and all of that stuff. I'm going to be discussing it, not in every single detail because there's not enough time for that, but as closely as I can without it being too much. So with that being said, let's get started. There are sections 5.1 to 5.6 in chapter 5. First, I want to start off with section 5.1. First page in the textbook is about eukaryotic cells, how they became, and what they are. So they were found around 2 to 3 billion years ago. The first evidence was found of the first cells by paleontologists. The concept of endosymbiosis is believed that bacterial and eukaryotic cells came from a cell named last common ancestor, abbreviated LCA. And then to the right there is a picture. It is just an example of endosymbiosis, which is basically just the eukaryote and then the archaea and bacteria, that process, how it goes along. And then its DNA is shown in the picture, and that is just an example, or one example of that. Bacterial and archaea cells were invaded and became part of the eukaryotic cell as the mitochondrion. The chloroplast in eukaryotic plant cells are photosynthetic bacteria. Still in section 5.1, the mitochondria and chloroplast share their own DNA along with the 70S ribosomes and 2-layer membranes. In medical microbiology, eukaryotes are the following. They are fungi, protozoa, and helminths. Protozoa are unicellular, while helminths are multicellular, but the eggs or larvae from the helminths are unicellular, not cellular. And then down below is just an image of what the eukaryotes look like. So it's showing bacteria, the helminths, the protozoa, fungi, all that stuff. Just a picture so we can kind of get an idea of how they look up close. Moving on to 5.2, which is the form and function and just appendages for flagella and cilia, getting into that. So the structure of eukaryotic cells contain the following. They have a cytoplasmic membrane, a nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton, and the glycolax. Some, but not all, have a cell wall, locomotor appendages, and chloroplast. Then bacteria starts with the outside and it goes inward of the cell. I added another image where each structure is located on the cell, where the cytoplasmic membrane is, the nucleus, and the rest of the following. It just shows where it is all located. Flagellum is long, sheathed, and it is a hollow microtubule that is about 10 times thicker. The 9+2 arrangement is a cross-section that shows 9 pairs of microtubules, which is a pattern of the flagella and cilia. Locomotion is when microtubules slide past one another, moving back and forth. And then the cilia, they're short, they're numerous, and they are in a single group of protozoa. Ciliated protozoa, they're in rows, and they also go back and forth. The glycolex is sometimes called the extracellular matrix. It's the outside boundary and provides protection, making the cells stick to other surfaces. The cell wall, fungal cell walls contain an inner layer of polysaccharide fibers, which is chitin or cellulose, with an outer layer of mixed glycans. Cytoplasmic membrane, a bilayer of phospholipids that have sterols. Sterols make eukaryotic membranes more stable. I just wanted to give some definitions of these terms just so we can get a better understanding. Now moving on to 5.3. 5.3 is really focused on the nucleus. So the nucleus is the control center, which is separated by the nuclear envelope, and it contains nucleoplasm. Nucleoplasm has fibers known as chromatin, which is the material of eukaryotic chromosomes. DNA molecules connect in varying histone proteins also. In the textbook, mitosis and meiosis is mentioned it was talking about the nucleus, so I just wanted to add in definitions of those two. So mitosis is duplicated chromosomes that are split to two equal daughter cells and they become visible. And then meiosis are sex cells, and they are made of only some cells go through this process. And then some more definitions and pictures to go along with those definitions. So the endoplasmic reticulum has two different types, the smooth and then the rough. So the smooth endoplasmic reticulum is a closed network that lacks ribosomes to function in nutrient processing. And then the rough endoplasmic reticulum are outer membranes of the nuclear envelope, and they span the network through the cytoplasm. And then that top picture is just the endoplasmic reticulum, and then shows what the smooth looks like and what the rough looks like. Then the Golgi apparatus, proteins are modified, then go to their destinations. They are associated with the endoplasmic reticulum and are comprised of polysaccharides and lipids. Then the second picture is just the Golgi apparatus and showing how it is connected with the endoplasmic reticulum. Mitochondria are round particles that are throughout cytoplasm. The folds are called cristae. Cristae carry electrons and aerobic respiration. They also contain enzymes. Chloroplasts are in algae and plant cells. They transform sunlight into chemical energy, which is known as photosynthesis. Ribosomes are the tiny particles in cytoplasm. Multiple ribosomes are called polyribosomes. And then cytoskeleton. The three main filaments of cytoskeleton are actin, intermediate, and microtubules. Then section 5.4 moves on to the fungi. So most of fungi are unicellular. However, the complex fungi is often multicellular. All fungi is heterotrophic. Morphological types of fungi are yeast and hyphae. I wanted to give a description of yeast and hyphae. So yeast is round and uses asexual reproduction. Then hyphae is long and in filamentous fungi or in molds. A lot of them get their substrates from dead animals and plants, which make them saprobes. They can also be parasites. Diseases are caused by fungi, and they will be called mycosis. The top image is just a foot infected with fungi, and then the bottom is just fruit that was contaminated by fungi. I thought it was important to give those examples. Then for 5.4, the reproductive strategies and spore formation for fungi. These are multiple types of spores. Sorry, there are multiple types of spores when it comes to fungi, and they are in charge of things like survival or producing genetic variation. Asexual spore formation has sporangiospores and kinetospores. Sporangiospores, they are successive cleavages called sporangium, and kinetospores are free spores. The other types of sexual spore formation, it is different genetic fungi that is combined with their material together. To the right is just an example of the multiple types of spores, the asexual spore formation, the sexual spore formation, shows some sporangium and kinetospores. Fungi affects humans and the environment that we live in every single day. They decompose organic matter and make a home in the human body. The three types of fungal disease are community-acquired infections, hospital-associated infections, and opportunist infections. They all weaken the human body in different but somehow the same ways. So you can really pick up fungi from anywhere and get it. Moving on to section 5.5, protostar kingdom called kingdom Protista, and the two categories are subkingdom algae and subkingdom protozoa. Algae are single-celled and filamentous form which can contain tissues and simple organs. They are photosynthetic protists. Protozoa are the reason for infections in humans, but only a few of these are to blame. Protozoan cells consist of cytoplasm and split into the ectoplasm and endoplasm. Some even have another layer which is referred to as the pellicle. We're still on 5.5 going over. So the feeding stage in protozoa are called trophozoites, and they keep them functional. A few protozoa groups only live in this trophozoite state, and they go back and forth between the state and the cyst state, which is going to be explained. So the cyst state is where they stop growing and stop the feeding process. To go along with this, ciliates engage in this process called conjugation. Conjugation is when two cells fuse for a temporary amount of time and swap their micronuclei. And then the picture beside of this is just protist, the process of that, and the different stages and states. Okay, moving on to section 5.6, which are the helminths. This section was kind of shorter, so I had to do the best I could getting information. So 5.6 goes over the helminths, which a helminth, the definition of it is just a worm, like tapeworm, flatworms, which basically is the same thing. A couple examples of this parasite is flatworm, which is phylum Platyhelminths, and roundworm, phylum Achinhelminths, or nematodes. Flatworms are thin and have a segmented body, while roundworms have a long and unsegmented body. There are two different groups of flatworms, cestodes and trematodes. Cestodes are considered to be like a skinny flat ribbon, and trematodes are rounded but also flat. The top image, you can see where it looks like a ribbon the way it's shaped, and then the image below is a trematode, and it is rounded but also flat. Life cycles of helminths, embryo, larval, and adult stages. In nematodes, the sexes are separated, but in trematodes, the sexes can share the same sexual organs. Humans can be infected by what they eat, touching their mouth, or drinking contaminated water. Then the image uploaded is from the textbook showing an example of how unclean hands can result in infection. That is the rest of my slideshow. I hope I added enough information. I think I did, but thank you.